Welcome! In this lesson, we build an intuitive picture of transmembrane signal transmission. External cues like hormones or forces are converted into intracellular signals by remodeling small functional membrane domains that act like signal amplifiers. Let's sketch a minimal membrane. We will focus on the inner leaflet, where special lipids recruit enzymes. These lipids are the anchors that bring the right proteins to the right place at the right time. First enzyme PI3K. It transforms PI4,5P2 into PI3,4,5P3. That new lipid, PI3,4,5P3, is a high-affinity anchor that recruits signaling proteins to the membrane. Second enzyme, PL-sigama. It cleaves lipids to make DAG and IP3. IP3 releases calcium inside the cell. DAG stays in the inner leaflet, while calcium rises in the cytosol. Together, they recruit and activate protein kinase C. Protein kinase C activation. PKC docks strongly when DAG is present and calcium reinforces a C2 domain that penetrates the membrane. This makes PKC a calcium-sensitive membrane-anchored kinase that can switch on many targets. Molecular switches like small GTP assays are activated at specific sites by GEF recruitment. When PI3K makes a local cloud of PI3,4,5P3, GEF proteins dock and flip switches like RAC1 to the on state, driving actin polymerization pulses. Resting cells keep PI3,4,5P3 extremely low, below 0.02%. The phosphatase PTEN removes the third phosphate, turning PI3,4,5P3 back to PI4,5P2. It binds membranes tightly with a C2 domain and leaves very slowly. Binding strength Kd about 1 nanomolar and off rate about 1/1000 per second. Calcium speeds up C2 off rates when absent. Together with PI3K, this tandem tunes sensitivity. How do proteins find rare PI3,4,5P3 among abundant PI4,5P2? The binding pocket can present a negatively charged sentinel near its entrance. PI4,5P2 is discouraged, but the more negative PI3,4,5P3 binds tightly, with Kd around 14 nanomolar for AKT. Removing the sentinel charge lets the pocket bind the wrong lipid, leading to always-on AKT and uncontrolled growth. Finally, receptor tyrosine kinases act as entry points and amplifiers. A pair of receptors dimerizes and phosphorylates tyrosines. These phosphotyrosines recruit scaffolds, and then PLC-gamma. PLC-gamma makes DAG and calcium, which anchor and activate PKC. Because of high affinity and complex cascades, responses can be triggered by very low hormone levels and last hours, reaching gene expression. Summary: membrane lipids like DAG and PI3,4,5P3 act as second messengers and anchors. PI3K and PTEN tune sensitivity. PLC-gamma creates DAG and calcium to activate PKC. Selective binding pockets use sentinel charges to find the right lipids. Receptor tyrosine kinases gather these modules into nanodomains that amplify faint external cues.